An algae bloom is a rapid increase or accumulation in the population of algae in a water system. Cyanobacteria blooms are often called blue-green algae. Algal blooms may occur in fresh water as well as marine environments. Typically, only a small number of phytoplankton species are involved, and some blooms may be recognized by discoloration of the water resulting from the high density of pigmented cells. When some of the nutrients that are in septic tanks leaks out and is mixed with stormwater runoff and then travels into bodies of water, the result is quite harmful. It can create algae blooms, which can disrupt aquatic ecosystems. Algae blooms are potentially toxic to humans and also pets, and have appeared in new waterways on the east end of Long Island very recently. Blue-green looking algae first appeared on Long Island in 2003 and was followed by red tide in 2006. Though many types of algae can form blooms, freshwater harmful algal blooms have the ability to produce toxins that are dangerous to other organisms, such as humans, dogs, and livestock. Most harmful algal blooms occur in warm, slow-moving eutrophic waters in mid-June through late September and are formed by cyanobacteria, which are now known to be photosynthetic bacteria. It is possible for blooms to appear quickly and form floating mats of various colors. However, not all blooms form mats on the surface. Some remain in the water column and discolor the water. While it is estimated that most blooms are toxic, it is difficult to predict exactly when or even if a bloom is producing toxins. A bloom of a certain type of cyanobacteria may or may not produce toxins, and the toxicity may change throughout the duration of the bloom. It takes a few days for blooms to be sampled and tested for toxicity, and by that time the toxicity may have changed. Furthermore, the harmful effects of blooms may even occur when a bloom is not visible. Algae blooms that occur in drinking water sources can produce compounds that lead to toxicity as well as taste and odor problems of the water. The toxins produced by cyanobacteria are the main concern during harmful algae blooms. They are produced within the cells and remain there until the cells break open, which may lead to the unpredictable toxicity of blooms. Normal water treatment processes can remove cyanobacteria and their toxins, but sometimes further steps are necessary when a cellular toxin concentration is high. The extracellular toxins are also more difficult to remove than the toxins within the cells. In aquatic environments, the toxins are broken down by heterotrophic bacteria, but during times of slow mixing, the toxins can build up because the bacteria can't get to the toxins as quickly. Microcystins are the most commonly occurring toxins. There are at least 60 microcystins known now, and the toxicities can be vastly different. Anatoxins are the cause of paralytic shellfish poisons are very poisonous. Lin-Bigyac toxin and aplasia toxins are both tumor promoters, and Langbia toxin A causes seaweed dermatitis. The toxins produced by cyanobacteria in algae blooms can also harm other aquatic organisms. The toxins produced by Cytomena hoffmanni and Fissurella musicola can inhibit photosynthetic electron transport. Anabaena flos aquae produces a toxin that paralyzes the green algae Chlamydomonas, and microcystins inhibit carbon fixation in other phytoplankton. Some of the techniques that can be used to control or remove blooms, such as algaecides, can increase the toxicity of the bloom. They also may be detrimental to organisms other than cyanobacteria and are generally not recommended. The most effective method is to reduce blooms by reducing the phosphorus loading of freshwater bodies. Although cyanobacteria are found in some of the oldest fossils and are natural to aquatic habitats, harmful algae blooms are increasing in frequency in the United States and across the world and are becoming more of a problem. Developing systems that can track and predict harmful algal blooms is one way to reduce this problem. In Massachusetts, scientists are experimenting with a sensor that can identify 3 types of dangerous microscopic algae by their genetic material and offer an early warning when they are detected in the water. In the Gulf of Mexico, NOAA's algae bloom forecasting system pulls together satellite imagery, information about water conditions gathered by weather buoys, and observations from scientists in the field to map blooms and predict how they will spread. Knowing when and where algae blooms are likely to occur can help scientists and public officials minimize harm to people and marine life. Learning more about the causes of blooms may ultimately also help us to prevent them.